And it's my great honor to have this opportunity to share our studies in transcription initiation. So in 1969, Professor Bob Rader identified the 3 eukaryotic RNA polymerases. We know that PAL2 can synthesize pre-messenger RNA and convert it into messenger RNA to guide protein synthesis. So this is the key step in central dogma. To make gene expression under process control in eukaryotic cells, so PAL2 alone could neither find promoter nor open the double-stranded DNA. So transcription initiation requires assembly of a pre-initiation complex called PIC, consisting of PAL2 and the general transcription factors called GTF. And TF2D is the first GTF to recognize core promoter and sequentially recruit PAL2 and other GTFs. The PIC complex has basal activity, and its activity could be further activated by TFs, mediator, and other co-activators. The GTFs was named originally from the purification procedures, but here I will show their functions that you may better understand their name. So TF2D functions in DNA binding, TPP loading, and PIC assembly. TF2A is an aid of TF2D, generating a DA complex, and the TF2B bridge PAL2 to DA complex, and the TF2F can facilitate PAL2 to bind the DA and 2B complexes. And PAL2 is the polymerases, and the TF2E can encircle DNA with PAL2 and recruit TF2H. TF2H is actually a final trigger for the initiation step. It has a helicase subunit can promote the DNA opening, and a kinase facilitate the CTD phosphorylation. CTD is the C-terminal domain of PAL2. It has a 52 heptide repeat in human cells, the 52 heptide repeats. So the phosphorylation of the serine residues at the 257 positions are critical for the regulation of transcription. And mediator is one of the most important co-activators. It can bridge PAL2 and TF interactions and facilitate transcription activation and CTD phosphorylation. So all the players are ready right now. So the PIC assembly has been nicely visualized by structural studies from labs of Kornberg, Kramer, and Noglas, and recently by Yanhui's lab. So in their study, they used the Tata box promoter and the Tata box binding protein to replace TF2D. So their studies made great contribution to the field. A remaining question focused on TF2D and promoter. So we know that human promoter are highly diversified in terms of sequence. And the majority of the human promoters are Tata-less promoters. They don't have Tata box. And TF2D consists of TBP and 13 TBP associated factors. The molecular weight is about 1.3 megadalton. It's a huge complex. And TF2D function is essentially required for all the PAL2 mediated transcription, and its function could not be replaced by TBP. So the first question we want to address is how TF2D support PIC assembly on different promoters. So we purified PAL2 and other GTFs to reconstitute PIC complex in different assembly states on different core promoters. So this complex was subject to cryo-EM structure studies. So by connecting over 20 structures, we can reconstitute the PIC complex assembly processes. So as shown in this cartoon, TF2D binds different promoters in a similar conformation. And the assembly diverges into two tracks. The Tata box promoter undergoes stepwise promoter deposition to PAL2, while the tatalase promoter is directly positioned above the PAL2 active site. The two tracks finally converge at the hollow PIC complex, providing a shared starting point for the following processes. So this parallel movie shows the difference of the two promoters. So this is a Tata box, this is a tatalase. So TF2D binds the promoter in a similar conformation. And the difference occurs at the early stage of PIC assembly. So TF2D binds the promoter because it has no Tata box. PAL2 and the TBP can move upward so that PAL2 can stabilize the promoter. While for Tata box promoter, it is positioned away from PAL2. And the TF2H makes the final push for the promoter to be positioned. But the final conformation of, on the two promoters, the PIC complex adopts in a similar conformation. So the Tata box binding protein could not bind tatalase promoter because it has no Tata box. But in the context of PIC complex, TBP can bind all type of promoters, regardless whether it contains Tata box or not. So the PIC complex assembly requires both TBP as a key promoter binding subunit and the TF2D as the essential organizing scaffold. So the TBP-generated promoter binding can prevent rotation or translocation of the upstream promoter during the initiation processes. The structural comparison also shows obvious difference between TBP-based PIC and our TF2D-based PIC. You can see the difference both in size and modular organization. For example, TF2D can support positioning of TF2H core and the TF2H kinase module on the surface of PAL2. So the kinase is positioned near the CTD region, and in vitro assay confirmed that increasing amount of TF2D can enhance CTD phosphorylation, but TBP showed very limited effect, indicating that TF2D serve as a critical scaffold to support the TF2H complex organization and CTD phosphorylation. So next part I will show you our unpublished data about visualization of the transcription initiation in action. The transcribing PAL2 could not stop at any specific position on promoter. So to capture the novel transcribing complex, we first assemble a PIC complex on 16 different GLAS promoters containing the G-stop region, the dinucleotide. So we next add ACU, but with the G replaced by a chain terminating analog. So in this reaction, PAL2 should stop at the G-stop position. The in vitro transcription assay confirmed the generation of nascent RNA with expected length. So we call this complex a transcribing complex with a number corresponding to the length of nascent RNA. So these complexes were subject to cryo-EM structural analysis respectively. So this is the cryo-EM maps of the overall structure, and the PAL2 with the closely associated GTFs were refined to about 3 angstrom resolution for model building and the structural analysis. We can also clearly see the RNA and the templated DNA within the active site. So these structures, according to their conformation, can be grouped into two classes. One is initially a transcribing complex from TC2 to TC9, and the early elongation complex from TC10 to TC17. In ITC complexes, you can see that GTFs remain bound to PAL2. The upstream promoter is bound by TBP and stabilized by TF2B, and the downstream promoter bound by TF2H, just similar as in the PIC complex. The upstream transcription bubble remains at the position of -12 relatively to the TSS. The downstream template strand continuously inserts into the active site, so generating a bubble extension from the 14 base nucleotides to 21 nucleotides. And you can also see the RNA length increases. In the EEC complex, the GTF dissociates from both upstream and downstream promoter regions, and the transcription bubble remains 11 nucleotides. A sharp transition occurs from TC9 to TC10. We call it ITC to EEC transition, involving the sharp bubble collapse and the GTF dissociation. An in vitro assay showed that in the absence of TF2H, the generation of RNA largely decreased, indicating a critical role of TF2H in initiation. So by comparing the structure of PIC and early ITC, we can propose a model of promoter opening. TF2H has a DNA translocase subunit, XPB. Upon ATP hydrolysis, XPB could pump the template strand color in yellow, about 13 nucleotides into the active site of PAL2. The upstream DNA is banded and stabilized by TBP and the TF2B and the TF2F. So you can imagine that both translocation of downstream promoter and the stabilization of the upstream promoter are essential for efficient promoter opening. This cross-sectional view of ITC shows that the active center merged with a narrow template channel. This is the template strand. The channel, we call it the channel T, formed by PAL2 and the TF2B colored in green, the B finger, B linker, and the B core region. And this channel is nearly sealed by the protein with two ends captured by RNA DNA hybrid and the DNA duplex of the upstream region. Well, in the EEC complex, because TF2B dissociates, the channel no longer exists, no longer sealed, but instead serve as a wide and short extension of the active site. So by comparing these structures, we hypothesize that the template strand accumulation in this channel and NTP-driven translocation of the DNA RNA make the final push to disrupt this channel, force the dissociation of TF2B, and promote the ITC-EEC transition. So this movie was generated by connecting these structures. So this panel shows the overall structure, this shows the omit structure, and this is nucleic acid. So TF2H pumps the DNA melting with the template strand inserted into the active site, generating a promoter opening complex. And upon the RNA synthesis, OC converts into an early ITC complex. The short RNA makes or fill base pair with the template strand, so the RNA is very highly flexible. From TC4 to TC9, the RNA becomes more stabilized, correlated with escape commitment. So because the upstream is fixed and the downstream template continuously inserted, and the upstream template get accumulated within this channel. And transition occurs when the RNA reaches to 10 nucleotides, force disruption of this channel T and the dissociation of TF2B. And the released template strand repair with the non-template strand, generating a sharp bubble collapse. So PAL2 continues move forward and escape the promoter. So this is pretty much what we learned from this study. So why does transcription initiation undergo such remarkable changes? So we have introduced that PAL2 by itself could not find promoter, could not open promoter. So initiation requires the GTF. However, the stability resulting from the multiple contacts among the GTF, PAL2, and promoter also creates obstacle for PAL2 to escape promoter, and therefore must eventually be disrupted. So in brief, early initiation requires the GTF to find promoter, open promoter, and support initial RNA synthesis, but the elongation complex requires GTF dissociation and the promoter escape. So I will quickly go through next two parts. The first one is about the mediator. Mediator can bridge TF to PAL2 and enhance transcription initiation and CTD phosphorylation. It has 26 subunits in human, and the molecular weight is about 1.5 megadalton. Our cryo-EM structure shows that mediator adopts an elongated conformation and packs on the surface of PIC complex. Mediator binds PAL2 CTD through a head-middle sandwich. So the structure will determine at near atomic resolution. You can clearly see the sequentially arrayed tyrosine residues of the CTD repeats. And the CTD extend from the mediator to the kinase. You can see this is kinase. So suggesting mediator can bring CTD to kinase for phosphorylation is consistent with previous biochemical analysis. So this is a brief working model. TF enriched mediator near the promoter region. Mediator binds unphosphorylated PAL2 and facilitates the PIC assembly. And the mediator also enhances consistent CTD phosphorylation. If CTD is sufficiently phosphorylated, mediator will dissociate from PAL2 and allows PAL2 to go into the elongation stage. So, and the mediator will support next round of PIC assembly and the transcription initiation. So we know that transcription actually occurs on chromatin. So in current model, the nucleosome on the core promoter will be ejected for PIC assembly. And the first downstream nucleosome is called +1 nucleosome. But in current model, the +1 nucleosome is functionally important. It's rich in epigenetic regulations and very important for the regulation of transcription. But there's no report how +1 nucleosome interacts with the PIC complex. It seems that it's positioned away from the machinery. The sequencing studies have shown that the +1 nucleosome is positioned it's well positioned relative to TSS. It's just about 40 base pair downstream of the TSS. So when we think about the role of the +1 nucleosome, we realize that the PIC-Mediator complex is actually very, very large. It consists of 76 subunits, 4.1 megadalton. It's even larger than the ribosome and comparable to HBV capsid. You can also get a feeling how big it is relative to nucleosome. So if we place the +1 nucleosome downstream, 40 BP downstream of the TSS, we suspect that the +1 nucleosome may generate direct contacts with this PIC-Mediator complex. So to test this hypothesis, we generate a template called the T40N. T means TSS and N means the nucleosome with 40 base pair separation. So we assemble the complex on this template and determine the structure. So as expected, the +1 nucleosome indeed makes multiple contacts with mediator and the PIC complex. So the in vitro assay also showed that the +1 nucleosome enhanced the transcription activity comparing to the naked DNA. So the commonly, the +1 nucleosome has been commonly believed serve as a barrier of transcription. So our study shows that the +1 nucleosome also serve as a hub to organize and modulate PIC-mediator complex on chromatin. So the positioning of this complex and the choice of TSS may depend not only on the sequence of the promoter, but also the position of the +1 nucleosome. And also, the role of the +1 nucleosome should also be considered for the following processes, including the initiation process or the capping, post-release and elongation complex assembly. With that, I would like to thank our talented group members. It's really a teamwork, especially for Xizhi Chen, who initiated this project about 8 years ago. And also thanks to funding support and the cryo-EM facilities. Thank you.